let's have a little bit more of a look at the digestion part of this process. So remember, this was step one, where we're taking in the dietary carbohydrate, lipid, and protein. So we have the digestion beginning in the mouth, and then we pass through the food into the stomach where it's further broken down, and then we have the completion of the digestion in the small intestine where we're actually able to absorb those small molecules, so our monosaccharides, amino acids, and the triacylglycerols through the small intestine into the bloodstream. So there are a lot of digestive enzymes that are involved in these processes, and we'll have a look at some of the specifics of these in each of our different digestion metabolism lectures that focuses on each of these three macromolecules. So not all of these three macromolecules have digestion occurring in the mouth, the stomach, and the small intestine. Some are predominantly more in the small intestine rather than the stomach, etc. So we'll have a look at some of these differences later on. So the important part of our digestion is the fact that we're able to absorb the broken down macromolecules, which are our simple macromolecules, through the intestinal wall into the bloodstream, and that's where we're going to be able to get these entering the cells. So these are going to be things like our amino acids, which we see here, our monosaccharides, and also the fatty acids and the glycerol that make up our triacylglycerol. Okay, so basically we use our digestion processes to break down the complex macromolecules into the smaller components, which can then be taken through the intestinal wall into the bloodstream and allows them to enter the cells where they can be used for metabolic reactions. So our acetyl group is actually formed within many metabolic reactions, and some of these will occur in the cytosol of the cell and others in the mitochondria. So depending on what is actually being used to break down, these reactions will be being performed in different parts of the cell. For example, if we're doing breakdown of fatty acids, then this actually occurs in the mitochondria, and this will create our acetyl group. So we can see here that glucose and fatty acids are oxidized to produce the acetyl group. So there's also a way we can make acetyl-CoA from the breakdown of protein, but it's simpler in glucose and fatty acids, so we're going to focus on that for the time being. So we have the generation of this acetyl group through the breakdown of our glucose and our fatty acids. So once we have the acetyl group formed, then we can attach the acetyl group to our coenzyme A, CoA, through the thiol. So we create this thioester bond that links the acetyl group and the CoA. So remember, the CoA has the thiol group and the acetyl group looks like this, with the carbonyl attached to the CH3, and then we have a spare bond here. So we need to form a new bond with something. So the way we can connect these two groups is by losing the hydrogen off the coenzyme A. Then we now have a coenzyme A, which has a sulfur that actually can accept another bond now because it's lost that hydrogen, which allows us to connect to the acetyl group, which we see here. So here's our CoA, and here is the acetyl group. And we can connect those two together because each of the carbon and the sulfur needs an extra bond. So this is the thioester bond that we are forming. So the important thing to remember about this acetyl group formation is basically that we can use the glucose and the fatty acids in oxidation reactions to create the acetyl group and then attach it to CoA. But just simply, if you realize that we create acetyl CoA from the breakdown of our carbohydrate and our fat through these metabolic reactions, then that is the overarching important message from this slide. So we create acetyl CoA from the breakdown of carbohydrate and also lipid. So once we have this acetyl CoA through these metabolic processes that have broken down our glucose and our fatty acids, then we're able to feed the acetyl CoA into the citric acid cycle. So that is the next step. So the citric acid cycle itself takes place in the mitochondria. So within the mitochondria, there's all of the enzymes that are required for the citric acid cycle. So what actually happens in the citric acid cycle is we are oxidizing the acetyl groups that are present on the acetyl CoA, and in doing this we're actually able to release carbon dioxide, energy, and our important coenzymes NADH and FADH2. So why is it that NADH and FADH2 are important products of the citric acid cycle? Are these the electron-rich or electron-poor versions? Remember, these ones are the electron and hydrogen-rich versions that can be taken to the electron transport chain. So the energy derived from doing the citric acid cycle is basically put into these reduced coenzymes. So we're putting in the hydrogen and the electrons that's resulting from oxidation of the acetyl CoA, and we're putting that into the NADH and FADH2. And we use these products in the electron transport chain. So we take both of these types of coenzymes that's produced in the citric acid cycle to the next step, which is the citric acid cycle. We do also have some of the energy that's produced in the citric acid cycle being lost as heat. And one of the byproducts is actually carbon dioxide. And that's where we get the carbon dioxide that we are exhaling. So we'll need to offload this carbon dioxide out of the cells back into the blood so it can be taken back to the lungs and we can breathe it out. Alright, so let's summarize what's important about the citric acid cycle. So citric acid cycle accepts acetyl CoA from protein, carbohydrate, and lipid metabolism. Okay, so we've created our acetyl CoA from doing metabolism of these macromolecules. We then oxidize the acetyl CoA, and the important product from this is our reduced coenzymes, which are NADH and also FADH2. And then we take these to the electron transport chain where we can create ATP. So basically we use the citric acid cycle to feed in the acetyl CoA that we gain from the breakdown of these macromolecules, which we then oxidize the acetyl CoA, and in the process of doing this we create these electron and hydrogen rich coenzymes which get taken to the electron transport chain to make ATP. So there's actually two processes that are occurring in the last step. So there's the electron transport chain and there's also oxidative phosphorylation. So the NADH and the FADH2 that come from the citric acid cycle, so remember we made these in the citric acid cycle, we offload these hydrogens and electrons into the electron transport chain, so they're being offloaded. And this is the first step towards creating ATP. So we actually have the electrons being passed on to different proteins within the electron transport chain, and eventually they get passed. The last step is to get passed onto oxygen, and by passing these electrons onto oxygen and also combining with some H+, we're actually able to convert oxygen to water. So this is actually why we actually require oxygen to live, because we need oxygen to perform this role in terms of being the final acceptor in the electron transport chain to ensure that we're able to keep making ATP. Without oxygen, then the electron transport chain stops, ATP production stops, and we die because we don't have any cellular energy to continue our cellular processes which keep us alive. So the other thing we get in the electron transport chain is we have the H+ being transported through to the intermembrane space of the mitochondria. So the reason why this is important is because we can use these H+ in a process that we call oxidative phosphorylation. So this is where we actually use these H+ to go through an enzyme called ATP synthase, and by the H+ flowing through this ATP synthase, it provides energy. So that allows us to perform this reaction. So we've got ADP plus phosphate, and we also have the energy provided by the H+ gradient, so the movement of our H+ through this enzyme, and that is actually able together to create ATP. So this is the final step in our metabolic process. We've taken in our macromolecules, we've broken them down, we've created the acetyl-CoA, we've fed acetyl-CoA into the citric acid cycle to create the electron-rich coenzymes, which we've then used in the electron transport chain to pass on electrons and couple this process to oxidative phosphorylation to make ATP using the energy from the H+ gradient. So basically we need the electrons being passed along in the electron transport chain to allow these H+ to move into the intermembrane space. Once they're in this space, we can then use them to pass through our ATP synthase enzyme, which facilitates the reaction we see below, where we have ADP and phosphate combining together to make ATP. But remember, we do need a large amount of energy for this, and that comes from our H+ gradient. So what is it that the electron-rich coenzymes NADH and FADH2 are actually providing for the electron transport chain? So they're actually providing the electrons for the electron transport chain and also H+, but the H+ aren't as important. So it's really the electrons that are being passed along, and this allows us to transport H+ into the intermembrane space, but they're not necessarily the same H+ that came off the electron-rich coenzymes. It'll make more sense once we look at this lecture. So I'll do a little drawing of this process just to try and bring this all together because it's a little bit tricky. Alright, so say we've got our NADH and we've got our electron transport chain, which is made up of these 4 proteins. So we give off the electrons into the electron transport chain, and then we pass these electrons through these different protein complexes, and then the last step is we add it onto oxygen to make water. So we actually need oxygen to take the electrons off the last component of the electron transport chain to allow another cycle of electrons to go through. So the reason why we need to move these electrons through in this way is because as we move electrons through, it's actually allowing us to put H plus into the intermembrane space at the top here. And then we can use this H plus to actually be pumped through another enzyme that we call ATP synthase. So the H plus goes through ATP synthase, and that's what allows us to do the ADP plus phosphate going to ATP. So the fusion of the coupling of the electron transport chain, which is what I've drawn on the left here, and oxidative phosphorylation, which is what I've drawn on the right here, is giving us ATP. So we will go into these processes extensively in one of the later lectures. So it's interesting to consider what would happen if the electron transport chain and or the oxidative phosphorylation was interrupted. So without oxygen, both the electron transport chain and oxidative phosphorylation would stop. And the reason for this is, as I told you on the previous slide, is that oxygen is needed to accept electrons from the electron transport chain to allow continuous cycles of the electron transport chain. So more electrons to be passed on to just keep the process going, essentially. So there is a component called cyanide that actually binds to one of the proteins within the electron transport chain and stops electrons being passed down the chain. So this has a similar effect to having no oxygen within the cells. So if we have no oxygen, then the electron transport chain stops, and if we have cyanide at a particular level, then the electron transport chain will also stop. So the consequence of these reactions stopping is the fact that ATP production will also stop, and therefore we don't have enough energy to keep the cells alive and to continue those processes that require energy. So this will eventually lead to death of the individual or the organism. So we talk about the citric acid cycle, electron transport chain, and oxidative phosphorylation together as the central metabolic pathway, because these are the reactions that are used to produce energy from our macromolecules. So another name for it is the common metabolic pathway. So you might see it called either the central metabolic pathway or the common metabolic pathway. So essentially we need all of these processes working together to make ATP, which is our cellular energy. So the other reason we can group these together is because no matter what type of macromolecule we're breaking down, we still need to use these three components to derive the majority of our ATP. So we can have carbohydrates, fats, and proteins all feeding into the citric acid cycle, which links to the electron transport chain and oxidative phosphorylation. And this is because each of these macromolecules can be broken down into those simpler molecules, and then we can create our acetyl group, which is CH3 C double bond O, and then we can connect that to our CoA, which feeds into the citric acid cycle. And the common metabolic pathway, all of these reactions actually take place in the mitochondria. So we've had a look at these 4 stages of the process to create ATP after we ingest food in more detail now. So we start off with ingesting the food in the form of fats, carbohydrates, and proteins. We then break them down to the simpler versions, and then we can use these in different metabolic reactions that we haven't talked about yet to create our acetyl group. And then we have the acetyl group combining with the CoA to make acetyl-CoA, which feeds into the citric acid cycle. And then we are creating these electron-rich coenzymes, which will then be used in the electron transport chain to donate electrons, which is coupled to the oxidative phosphorylation, which will create ATP. So you can think of the original inputs here being the fats, carbohydrates, and the proteins, and the ultimate output is the ATP, which is our cellular energy, which we require to keep us alive. So I think we've basically covered most of that in the last few summary slides. So just remembering back to some of the structures of our coenzymes. So we had the coenzymes having a combination of vitamin derivatives like our flavin and our nicotinamide, and we have them bound to other sugars and also the ADP. So we had our coenzyme being made up of these three components. We had the vitamin derivative, then we had a sugar, and then we had ADP. So remember, ATP has the same structure as the nucleotide. We've got the sugar, the nitrogen containing base, and we've also got one or more of these phosphates. So if we had adenine with 3 phosphates within this structure, then that was where we had ATP, whereas if we had adenine with 2 phosphates, then that was ADP, and adenine with a single phosphate was AMP. So remember, each of these also has the sugar bound in the center. And the number of the phosphoanhydride bonds that our ATP, ADP, and AMP variants have is 1 less than the number of phosphates. And remember, by breaking this bond, that's what gives us the large amount of energy which can be used to fuel the cellular processes. So I'll see you next time for the first lecture in our carbohydrate series.